Bismillahirrahmanirrahim. Assalamu alaikum. Thank you very much for this invitation to present in Third International Conference on Applied Zoology 2020. I am going to present on genome editing in dengue vector Aedes aegypti for its genetic control using CRISPR-Cas9 system. As you see, I am Dr. Kashif Zahoor. I am working as an assistant professor in the Department of Zoology, Government College, University of Islamabad. Here, it's my presentation scheme. First, there will be some slides about this identification of mosquitoes, its life cycle, and then its management strategies. And then I will focus on this genetic control using various techniques nowadays. And then I will go to my topic CRISPR-Cas9 system in and its applications, and then using in this Aedes aegypti. Then I will present the research plan. Here in Quran Pak, Allah Ta'ala mentioned in Surah Baqarah 26 ayat, indeed Allah is not timid to present an example that of a mosquito or what is smaller than that any of. So it shows the importance of mosquitoes. Even a minute insect like mosquito, it has an importance. Allah Ta'ala mentioned in this in his Kalam, Quran Majid. So mosquitoes, they belong to order Diptera. Diptera, they are true flies. They have two wings. Technically, they have four wings, but two wings, they are rudimentary. They are halteres. They are distributed all over the world. Globally, they are everywhere. There are more than 3,400 species, including 37 genera, and all of these species of mosquitoes, they are included in one family. They are small insects, fragile, and 3 to 6 millimeter long, and they are everywhere. They have certain flight range from 20 to 30 kilometer without any rest. And females, they are blood sucking. Males, they feed on juices of flowers and fruits all the day. They are outside, and females, they only feed on blood for their egg laying. So there are certain stimuli which can attract females. They are sweat glands, fat contents, heat, carbon dioxide, body color, uh, color of clothes, and you see heat. As I told you, there are 3,400 species of mosquitoes. About 300 mosquitoes, around about 10%, they are disease causing. And I told you these female mosquitoes, they feed on blood, and males, they feed on juices. So only females, they are responsible for the transfer of diseases. Aedes aegypti is responsible for hundreds of millions of human infections: chikungunya, yellow fever, dengue, and Zika viruses. Here you see there these three genera: Anopheles, Aedes, and Culex. And you can see even from egg stage to larva and pupa and to the adult stage, we have differences. Eggs, they are different. In Aedes aegypti, they are single. In Anopheles, they have satellite pattern, and in Culex, they has raft-like structure. And in Aedes larvae, we have siphon. This is, I circle this. This is a siphon for respiration. This is horizontal to the water surface. In there, it's absent in Anopheles, and in Culex it has a certain angle, making an angle with the water surface. And I did not show you the here mosquito pupae. Here you see the head structure. You can differentiate between males and females, and these are adults. Even the body structure, the stature, or the sitting position, you can identify at general level. The central portion is for Aedes. Here you see there are males and females. Males, they have bushy antennae. This is called plumose, more robust and more feathery. And females, they have pilose antennae. They have less bushy and less feathery antennae. You can differentiate, even they are flying. You can differentiate between these two genders. Here, this is life cycle. Normally, one female mosquito, it lays more than 100 and around about 200 eggs. There are 4 to 5 batches. These eggs, they are hatched in 48 hours. After 2 days, it depends. In insects, it depends upon temperature, climatic conditions. If the or temperature is low, developmental is delayed. So this is under optimum conditions. They have 48 hours for hatching. Then there are 4 to 5 days, or normally 4 days for instars. There are 4 instars in Aedes aegypti, and then after fourth instar, they develop or they metamorphose to pupae. Pupae, it depends upon temperature. So round about at normal conditions, they take 2 days to emerge into adults. So in the right panel, you see there are certain instars in our lab. We rear these mosquitoes, and interesting thing is this: in Aedes aegypti or in mosquitoes, even pupae they move. So this is coma shape pupae. In this right panel, you can see. With respect to my topic of discussion or topic of presentation, and generally on dengue mosquito, we have these 2 chapters. On left panel, this is dengue fever, a general perspective. This is intake open. We published this in a few years back, and in recently it is going to be online with this CRISPR-Cas system in insects. There is a brief and very good information about this CRISPR-Cas system in mosquitoes in this chapter, in this book. This is the 4th chapter about this CRISPR system in insects, and particularly with mosquitoes. If anyone is interested to read more, he can go through this. So let's come to the point on this control measure. There are certain control measures for the control of mosquitoes. First one is the chemical control, which is obviously most quick and robust control, and biological control using certain predators and parasites. It's not very much reported in mosquitoes, but normally for insects we use this. There is a mechanical control. There are certain electric rackets and certain other control measures. They are common but not very effective. There are culture control to eliminate the breeding places, and there are physical control measures, using screens and using these certain chemicals in whitewash, and they are putting on these walls. And there is genetic control. We will focus on the genetic control. So up to this, the most obvious method for the control of mosquitoes is considered as chemical control. Last year, we submitted this article about the uh, molecular analysis of, this is serotyping analysis and molecular analysis of Aedes aegypti. We did this in Khyber Pakhtunkhwa. One of my student of MPhil, he did this work. We actually got 15 Aedes aegypti population samples, and then out of these we found only 2 positives for DEN2. Even out of these 4 serotypes, we found DEN2. It means if we are using chemical control, chemical or insecticides everywhere, this is not a good choice. So chemical control as everywhere is useless. As you see, out of 15 only we found 2 positives. So 13 sites with the heavy use of chemicals, with the wastage of money, this is useless. So there are many disadvantages of chemical control. We can summarize in toxicity to non-target organisms. If we are going to control mosquitoes, we are going to control every organism, or we are uh, producing toxicity to other organisms, non-target organisms, predators, and other beneficial organisms, beneficial insects. The second is the biomagnification. If we are putting this insecticide into the environment, they go to the soil, to water, and then water to the animals, to mammals, and then from milk to human beings. So there is a biomagnification which is very much injurious to human health and of course to the environment. And the other thing is very much interesting, very much alarming nowadays. This is genetic resistance in mosquitoes or in other insects. One of my student, she did this on this genetic resistance. There is a gene, knockdown resistance gene. We found heterozygous and both homozygous mosquitoes in and housefly populations in Punjab. So if there is resistant strains, they are in any given environment, we cannot have any control. So we are wasting our chemicals. So you can see chemical control gives good results, but it is not. It is the wastage of money. It is the toxic to environment and to human health as well. So there remains the strategy genetic control, which we can focus. This is environment friendly. This is targeted, and it gives promising results. So there are certain techniques which are being used in 90s and then in 2000, 21st century in the last decade. There was once one technique, RNAi, gene silencing through RNAi. So idea is this to generate double-stranded RNA with about more than 20 nucleotides, 21, 22 or 23 nucleotides. They target the gene of study, which is going to be to silenced. So there are certain drawbacks for this technique. This off targets. For example, this is a 500 base pair of gene, and you are targeting by using this 22 nucleotides. You can have many more of targets. So this is research point of view. This is from this point of view, it can be possible, but in highly organized eukaryotes like human beings, it is not very much commonly used, but it's in use. The other techniques is sterile technique, SIT normally called. There is a Oxitec company in UK which is using these mosquitoes and SIT mosquitoes, and it's I think in Malaysia, Singapore, or in Thailand, and either in Brazil they use this technique. This is very much effective technique, but it has certain drawbacks as much as has been reported. This is zinc finger nucleases, which is parallel to this genetic control techniques with other techniques, but it is not very much focused or targeted. There are certain drawbacks for this technique, difficult, and there is talent technique, which is also difficult, and there are targets of targets also reported. Despite of having many these genetic control techniques, there is another technique which is called CRISPR-Cas system. This is reported technically. It is a adaptive immune system reported in bacteria. So it has been borrowed from bacteria to the to other organisms. So recently it has been uh, being worked in Drosophila and introduced in other organisms. So in Aedes aegypti, it is also reported to be used with certain genes. CRISPR is basically abbreviated to this name, cluster regulatory interspace short palindromic repeats. It's carried by certain Cas9 endonuclease, which targets a certain DNA, certain gene you are going to edit or you are going to modify, you are going to silence. So using this endonuclease Cas9, there is more specificity, more targeted, more focused result, and this is environmental friendly. Environmental Protection Agency of USA, they spotted this and they say that this is safe as compared to the control techniques. So this system relies on RNA DNA based pairing to generate targeting specificity, resulting in efficient and flexible genome editing in organisms. So here This is the mechanism of CRISPR-Cas system. Here you can see pink color, this is purple color, Cas9, this is a endonuclease, and this guide RNA. This is basically a guide RNA of more than 100 nucleotide base pairs. You, you have to target a certain gene, so you design this guide RNA and along with other components, and this is with Cas9 endonuclease you introduce into the cells, and this Cas9 causes double stranded cut in the DNA. So this DNA, then there are certain system you can see afterwards. So basically you targeted or you focus the change or amendment or mutation in your into your target. This is more specific and more focused one. So this is a next gene which is called male determining factor. Interestingly, normally we have XY chromosome system, but in Aedes aegypti there is no XY system. There is a certain segment or there is the certain repetitive sequence of DNA which is called male determining factor. This is M factor. In M factor there is a gene that is called NIX. During certain embryonic stages in developmental stages, you can see in the egg stage, if this sequence is expressed in embryonic stage, if it is expressed, this is going to be a male. If it is not expressed, there is no repetitive sequence, so this is female. So you can see in left panel, in male this is a PCR of NIX gene. So you can see in males it is expressed, but in females it is not expressed. And this is the below in left panel you can see from 0 to 12 hour embryonic stages, it is going to be expressed in the fourth hour in the development of embryonic development during egg stage. So in the right panel upper, you see if if NIX is expressed in females, they developed maleness in its body parts, and if it is absent in males, so there is no male genitalia developed in males. So this is the M factor from which we can learn that if we express NIX in females, they develop into males, and if it is not expressed in males, so they are not no more males. NIX cytologically it is present on chromosome 1. It is not XY. So there are certain information. Sex differentiation is under control of array of regulatory genetic network of in insects. This is there are certain genes which are in this genetic or sexual pathway. There is sex lethal, transformer, double sex, and fruitless. They are reported in insects. This is the whole picture of this genetic pathway or genetic sexual pathway in insects. NIX basically is controlling, which is reported, is controlling the expression of certain maleness and femaleness in mosquitoes, Aedes aegypti. So under NIX, double sex can produce both the phenotype of males and females. Here and fruitless, this is fruitless means there is no progeny if it is expressed. So this is the whole picture shows you certain genes that which are present in the uh, sex determining pathway in insects. So you see double sex is epistatic, this sex lethal is epistatic double sex, and transformer is upstream of this double sex and fruitless, and NIX is somewhere. So this is the whole picture we draw from certain research papers and articles and from this website from protein interactions. So this is the main, we found this certain genes which are involved in the sex determination pathway in insects. So we are going to focus on this NIX and what does it do with this sex lethal and transformer genes. So we don't know up to this time, but we are going to explore this to know more about the sexual pathway to generate maleness in females. So DSX and fruitless, double sex and fruitless, they give rise to both males and females. And if injection of sex lethal fruitless and genes in transformer, these are certain informations available in literature reported in Albopictus, Aedes albopictus, and Drosula melanogaster. So we found through this GS2 hybrid assay a physical partner of these nicks, and we are going to focus on this. And when we found technically there are two genes, I mentioned this in the circle, sex lethal and transformer, and these two, they are physically linked to other partners, of course, and we will use these two genes to have amendments, or they to have been changed in future in Aedes aegypti. And then we will have these mosquitoes, we cross these, and we will be looking for a good line, a good strain in the lab to control uh, genetically control Aedes aegypti. So the overall objectives, there is one major objective of this study, genetically to generate genetically edited Aedes aegypti mosquitoes for the genetic control through this CRISPR-Cas system, CRISPR-Cas9 system. And there are certain sub objectives. We will use this single gene and multiple genes, and we will use the other genes like NICS and double sex and fruitless to know the whole picture of the genetic pathway. So we have these sub-objectives to perform single and multiplex gene editing for male determining factor, NICS, sex lethal, transformer, transformer 2, DSX, double sex, fruitless genes, and other physically linked partners involved in sex differentiation pathway using yeast hybrid assay to characterize NICS, sex lethal, transformer, transformer 2, the double sex, fruitless genes, and other physically linked partners involved in sex differentiation pathway. And we will be able to procure the genetically edited Aedes aegypti mosquito strains and to monitor their maleness in characteristics in females. So we have this plan of work. First, we have this collection of mosquitoes. We have this strain. We are rearing this mosquito strain, and after this, we have this isogenic. This is free of insecticide, free of pesticide, free of this. This is having isogenic background in this strain. We will collect these eggs, and then meantime we will design a single guide, this guide RNA, and this guide RNA we will clone this into plasmid, and then this plasmid will be going to be injected into embryos. These embryos, they will give rise to adults. This we have this G naught population. This G naught population, we will be going to check the expression of double sex, fruitless, NICS, and depending upon the gene which we are going to change, or then we have this PCR to characterize this if it is present or overexpressed or not expressed. It depends, and then we will quantify this messenger RNA using qPCR, and then we will be characterizing protein level expression if it is expressed at which level. So we will use this spectrum plotting technique. So after this, we will be able to generate a specific line of maleness in females, inshallah. So this, as I told you, this is we have collection of Aedes aegypti mosquito, and one strain we will be procuring from abroad, from USA, just to have a control in parallel. And we have this line to be used for microinjection. This there is a plasmid we are going to construct using these genes. The candidate genes are Aedes aegypti sex lethal, Aedes aegypti transformer, Aedes aegypti nix, Aedes aegypti double sex, and Aedes aegypti fruitless. All of these will be used singly and in combination and inserted into this plasmid. Then we will use this for this microinjection. Then we, as I told you, we use these messenger RNA will be transcribed using these PML M36313 will be obtained from a gene. This is already reported. So single guide RNA will be designed as already reported the procedures. So we will use this 4 hours embryo. We will get eggs, and then we will have this microinjection of this plasmid into these eggs. They will give rise to adults, and after certain stages, and then we will monitor all these stages, and after this we will be looking for this strain. I would like to mention here there are certain microinjection systems available from Appendorf, which is the best one, and also certain uh, microinjections or small needles available which are being used by these sugar patients or diabetic patients. They can also be used for the microinjection system. They are also been reported in literature. So we will be looking for this microinjection system, as well as if it's available a very minute needle for this plasma injection. Then we will be using this PCR technique, and then we will sequence the DNA if there is expression of this specific DNA, specific gene. We are going to change this, edit this in the genome of Aedes aegypti, and then we will be characterizing or quantifying the messenger RNA level using qPCR. For this proteins, protein quantification or protein expression level, we will be looking for this Western blot technique, and then after this we will having this line, first line, then we will have in intercross, and after crossing this we will have a G1, and we will inshallah establish this line in future in our lab. These lines, how many larvae they are being hatched from the eggs. We will be counting these larvae, and from these larvae how many they survive, and because after mutation or after amendment, or you can say after genetic genome editing, they have certain reports, they have less survival. The survival percentage is less. So we will be going to observe or record all these hatching and larval development stages, and how many they are going to emerge in adults. So all these, they will be, the data will be observed, and after we will use suitable uh, statistical design like this Takis HSD test, honest significant difference test, or SPSS, so we can have this data analyzed by the statistical means. So I show you the certain results. We up to this time we have this. So you see, I have this circle. I already showed you this slide. So what is the interaction between Nix and sex lethal and Nix with this transformer? Actually, in Aedes aegypti, there is no transformer like gene another that is being reported in Drosophila. So this is called transformer 2. We are looking now Nix in interaction with this sex lethal and Nix in interaction with this transformer. We will be looking for the expression of this double sex and fruitless in these lines after this. So this is a huge work. So we have started this one using this transformer 2. This transformer 2 is located in chromosome 2. It is a gene sequence can be retrieved from NCBI. with the accession ID I mentioned here, KF227901.1, start codon and stop codon, mature codon sequence, and 5. Also, we have also retrieved this. We have designed certain knockout experiments. So up to this time, we have this transformer to guide RNAs. We have designed two guide RNAs. Uh, so these with these guide RNAs, we selected these because with certain specific characters. We focus on this because with these two guide RNAs, there should be no off targets, and they will have good results. Or I mentioned you, if you can see here, there are certain specific sequence in uh, this yellow color sequence here. I forgot to mention here, this is called PAM sequence. So this sequence is very much very interesting and very much important. This sequence is recognized by uh, Cas9 endonuclease. So we designed these two guide RNAs having these PAM sequences. So this sequence is very much specific. So we have to look for. There are too many guide RNAs you can generate from this the whole gene of interest. So we selected two with no off targets. So up to this time, after this guide RNA, we have also generated our plasmid with this uh, for this gene transformer gene, and we are working on this up to this time. This is a huge work for looking for the whole gene, and then after from these genes you are looking for PAM sequences, and then you are going for to look for off targets, and then you select this. So with this transformer we have this selected this sequence and guide RNAs and this plasmid, and this plasmid is ready, and we are going to make a micro injection using this plasma, inshallah. So after this we will have results, inshallah, in few months. Then we will be able to do with side by side with other sex lethal and fruitless and double sex, and also with nix, and we will having a good story about this the sex differentiation pathway. So these are certain references if you are going to look for certain material. Thank you very much.